Thanks to everyone for coming. Some chats coming in too. Thanks for the hellos, Chris and Meg. So today at the AgBio meeting, we're going to talk about the GFF3 format and all of the joys and tribulations that come with it. Here, let me get into presenter mode. This is Monica, by the way. Hi, everyone. Thanks for coming. And I'm in presenter mode. Can you guys see my screen? Does that look okay?
Yeah.
All right, thank you. But before we dive into that, just a brief update on what's going on with AgBio and the upcoming conference calls. So on April 8th, we usually have these calls the first Wednesday of the month, but next month we're gonna have to postpone by a week. It'll be Dr. Mara Devar from CGIAR talking about the Guardian platform, which is a newish platform for fair data sharing as well. On May 6th, we're going to have a group discussion on pangenomes, which is a topic that a lot of people have been wanting to bounce some ideas back and forth about. And in June, Dr. Julie Dunning-Hotep from University of Maryland will be talking about her research parasite award, so secondary data usage analysis. She also does really cool stuff on lateral gene transfer. So with that, I was going to show the agenda, and I'll try to, hang on. I wanted to post this note document in here. We're going to be taking some notes as this is going and recording this. And oh, Jacqueline, you're recording, awesome. So I'll post this link into the chat. Sorry, stop sharing. We'll be trying to take notes as things are going along. All right, let me share my screen again. And before I start, yeah, it looks like we have Scott and a number of people on who have volunteered to share their experiences. So we'll go and do that. Hang on one second. Oh, it is sharing. I'm having technical issues. All right. Okay, so what are we going to talk about today? I was going to briefly introduce what the GFF3 format is for the uninitiated. And just briefly discuss some of the history of its uses, but also its drawbacks. And then the idea here, we don't want to have an hour where we just whine about our problems with GFF3. That might be fun, but it's not very productive. So the idea here is, do we have enough people in AgBio data sharing the same problems with this very commonly used format? That we could work together to at least solve a problem or two that we're commonly having with it. And somebody should probably mute their phone because I'm hearing a lot of echo. So we have a couple people who volunteered, or who we volunteered to give some examples. Your name is here. I'm not trying to really call you out. If you don't have anything prepared, don't worry. We can just move on. And if you have something that you want to share and your name isn't on here, that's awesome too. We'll have about half an hour to discuss those examples. And then once we've talked a bit about what our problems are, what we'd really like to have is some discussion. What are common issues that we as a group could aim to fix to work together? So best practices to solve a particular problem, like agreement on the structural annotation of a protein coding gene, how we represent that in GFF3 format so that we can all slurp it into our respective databases, that kind of thing. Or maybe a page on the AgBio data website where we have toolkits to fix common problems. And again, here's the gathering document for notes. So somebody chat. Okay, thanks, Meg. Okay, so first I was gonna give people an idea of the GFF3 format. This is a lot of text, you don't need to read it. I just wanted to make clear that there is a formal specification that's here up on the Sequence Ontology GitHub site. And when you look at the site, at the specification, at the very top, there's a paragraph or so written by Lincoln Stein, where he gets, in my interpretation, a little bit salty about the justification for having the GFF3 format. But basically the idea is bioinformaticists, they want a flat file format that describes genomic features, so annotations in structural and functional format. And there's been a ton of ad hoc tab delimited flat file formats, the GTF came to mind, or the format before GFF3, which was GFF 1 and 2. So these darn bioinformaticists want something that they can modify with a text editor and process with grep. What motivated the rise of the GFF3 format was to improve the GFF format, to give it some structure. So there's already been a bit of history with GFF in getting it to work for the larger community, and there's been improvements over time, but we're seeing that there might be some things that could be improved upon for GFF. So here, just to give you a brief overview of what we do with it. So here's, again, from the sequence ontology specification site for GFF3, here's their example of the canonical gene, where you have your 10 kb of nucleotide sequence describing coordinate space. You have your gene in that coordinate space here called Eden, 3 alternately spliced mRNAs belonging to that gene, the third one having a modified transcriptional start site, and you have a promoter for it. So this is kind of a, what can we call a gene? What's the classical case of a standard protein coding gene? And so we describe this in this beautiful tab limited text format. It's GFF3 has 9 columns. Sorry to bore those of you who know this by heart already, just wanted to point it out. And those 9 columns describe kind of the structural annotations. So what are the coordinates on the protein or on the genomic sequence that would give rise to the mRNA, the coding sequence, the protein sequence, but also here in this column 9, where a lot of the GFF3 magic happens, the functional annotation. So what's its ID, what's its name? There's a lot of parent relationship going on where the different sequence ontology feature types, gene, mRNA, their relationships are shown with each other. And the canonical gene in GFF3 has guidelines on how to structure a protein coding gene, like you would have a gene parent, mRNAs, exons, and coding sequences, CDS features that describe where those things start and stop on the GFF, on the genome, or the reference sequence. But as people who have worked with this format probably know, not all programs or people actually do that. They might use an mRNA as their parent feature with CDS as child features and no exons. And then when you use downstream programs to slip that in, they might make assumptions about what the format should be instead, and then they break. So some common issues, just to start things off, that we might be experiencing in AgBioData is interoperability. So as I mentioned, you make some assumptions about how your protein coding gene is formatted, and you try to ingest it into your database with your scripts, and it breaks because the specification is loose and somebody decided to do things a different way that fit their use case better. Using another group's GFF3 and a standard tool for your database, same kind of thing. One thing that we've had with some of our users complain about with insect stuff is if I5K provides a GFF3 file and AphidBase does, they're not always formatted the same. Can't you guys agree on at least giving us the same GFF3 files so we don't have to reformat the insect GFF3 files to do their processing, kind of thing. One of my pet peeves is not interpreting the CDS phase column correctly, which can break the protein coding sequence. They're easy to manipulate, and therefore they're easy to break. So often we see bad GFFs that just don't work at all because they were manipulated by a human. And just to note too that there are 2 types of annotation represented in GFF3 files that might require 2 separate discussions as we go along. Structural annotations that describe how the gene is structured. So that describe what the resulting gene mRNA protein sequence would be, and functional annotations that describe any metadata about that annotation, so its name, etc. Right, so now I was going to open it up to AgBioData members to get some idea about what the common problems are that we're experiencing. And Scott Kane has graciously volunteered, or maybe we strong-armed him into giving a little overview of what the Alliance for Genome Resources has been doing for the GFF3 format. I think you guys have been struggling with similar problems. So Scott, I was going to stop sharing and allow you to share. Is that okay with you, or do you want to do it differently? Yeah, I could also.
No, I'll share. That's fine.
Sounds good. I'll stop. You start.
Okay. see. There we go. All right, you can see my screen?
Yes, got it.
Okay, terrific. Very good. Okay, so I am the Gmod project coordinator. I've been doing that since 2002, so it's been a while. I'm also a developer at Wormbase and a developer at the Alliance of Genome Resources, and the leader of the Genome Futures Working Group. So just a quick background, the Alliance of Genome Resources is an NIH-funded project where the goal is to create a single resource for helping researchers identify model resources for human health research. So it's basically got all of the big NIH-funded model organism databases: yeast, fruit fly, worm, zebrafish, mouse, rat, and also the Gene Ontology Group is part of AGR as well. So as you might imagine, there's a lot of cultural inertia associated with each one of these model organism databases because they've been around for a very long time. Most of them since before the year 2000. So getting them to work together is not so easy, even though we all want to. We have a lot of things, a lot of barriers to get through to make it work. So what we have are a variety of working groups centered around a particular topic. So we have anatomy, expression, so biology topics, plus computer topics like user interface and data wrangling, which is where a lot of that integration stuff happens. You know, basically they're at each model organism database, there's one person who's designated the data quartermaster who's responsible for coordinating with the other data quartermasters for pulling data in. And then finally, there's the Genome Futures Working Group, which is the one that I'm the leader of. And so that's basically creating a JBrowse for the site that encompasses all the species, plus creating in-page widgets that we that are publicly available. So if you wanted to put one in your model organism database, you could do that too. So just very briefly, this is going to be a very short talk.
Yeah, Scott, before you move on, sorry, are you
yeah.
switching your slides? Because we're still looking at the title slide.
Oh no. Oh. Okay, let me, thanks for pointing that out. I think I know what to do. I need to stop sharing and then restart here.
All right, now we see attempt at a standard.
Attempt at a standard, very good. Okay, right. I think that'll work. Okay, so, so the Alliance has been around for, I feel like 3 years. That's kind of the right ballpark of time. And so we're going to do our third kind of big release. So version 3 is coming out in the next couple of weeks. So we've gone through a few iterations of this since then. So basically for the first release, which was a few years ago, our attempt at a standard was basically to ignore everything except protein coding genes. And so the way to do that was first to agree on how to represent the central dogma in GFF and in terms of SO terms. And so, and you would think that doesn't seem like that should be a very difficult thing to get people to agree on, but again, a lot of cultural inertia. So, so basically kind of one of the very big important things was that the top level item is gene, and it is not anything else. So it's not protein coding gene or anything else like that. It is gene specifically, so that we would all have, you know, exactly the same top level sequence ontology type. And then its children, its direct children would only be mRNA, since we're talking about protein coding genes. That's the only thing that's allowed. So there's no other transcript types you're allowed to use. And then CDS features are absolutely required, but other types, you know, other components of mRNA are not required, but they're allowed to exist there. So exon and the 3' and 5' UTRs, things like that. Those can be there, but they aren't required. And another thing that we did fairly early on was decide there would be no restriction on what's in column 2, which is referred to as the source. So source was intended to be kind of a free text, whatever you think should go there can go there, and we decided to leave it that way. Because at some of the mods, the source is the mod itself. So Wormbase and Flybase, for instance, they're the source. But at some other mods, the source is Ensemble or NCBI, things like that. Okay. There we go. So other standard items we agreed to at that time was that there would be, I think we required one tag then. Okay, so the first thing is there's a required tag for gene features. They would all have a Curie tag, which is a URI. Basically, it's a resource-wide universal identifier for the features that were going into the Alliance. And then there were other kind of several items that you would kind of consider convention that we specified specifically how to deal with them. So for instance, dealing with CDS features, in GFF, you can group them just by parent tags, basically saying, well, this CDS, all these CDS lines belong to this transcript, therefore they all go together. So that's one convention. And we decided to leave that as explicitly how you can do it. You can do it most of the time. But if you have a gene where the transcript results in, excuse me, we're in a small apartment and people are moving around. So anyway, so yeah, if you have a transcript that results in more than one CDS, then you are required to explicitly give them IDs so that you can distinguish between them. Also, we required that there be a comment near the top indicating what assembly the GFF is derived from. Then, okay, so after that first release of the Alliance website, we generalized to non-coding genes. So basically, this was why in the beginning we said the top level is going to be gene. We're going to keep it so that the top level thing is always gene, but the transcript type that is the child of it is then a transcript type that is specific to what sort of gene that is. So sequence ontology terms like tRNA and rRNA and SNO RNA, all those things, that's where it would identify, it would specifically say what sort of thing it was. Then also a new tag would be required in the 9th column for the gene line that was so_term_name, where the value of that is the sequence ontology term for the most correct type of gene. And so we kind of did this so that one, building JBrowse would be easy because then we just have gene. We say, okay, this is the track that's all the genes, but then if we want to do something specific to say a glyph, then we could look in column 9 and say, oh, well, this is a protein coding gene, whereas this is a tRNA. So if we want to color them differently or draw different glyphs, things like that.
Scott, can I ask,
yeah.
Why didn't you take the transcript? So like you have your gene parent feature, and then presumably a transcript is usually the direct child feature of the gene. So why didn't you take the sequence ontology type for that one to describe what the biological function of that transcript is?
Wait, I'm not sure I understand your question, because I think so we did do, so for the transcript line, so the thing that would, we did use the sequence ontology term that's specific for that type of transcript.
So, but that's,
I think is what you just said.
So yeah, right. So, but you found the need that in addition to that, you also required a, oh, okay. So you put that on the gene level, so you don't need to try to traverse down. Okay, got it.
Right. So we don't have to. So if we're doing something in code, we don't have to look up or down to say what type of gene is this. It's just there.
Okay. This is Chris Elsick. Can I ask a question related to this?
Sure.
So what if like so like NCBI will have a gene locus that has both protein coding transcripts and long non-coding transcripts.
Yeah.
And so that always gives us trouble, and we have to, especially with JBrowse, and we have to separate them out. And and then so on like the way Ensemble, when they do their GFF, they indicate it at the transcript level. I mean, okay, so would you, let's say one of the NCBI genes that have both, would you just call that a protein coding gene and then let it have some non-coding transcripts?
Yes. So that is actually the very next sentence, which is when a gene has both coding and non-coding transcripts, the value should be protein coding gene. And
can I ask?
Okay. Sorry.
Yeah. So several years ago, I wrote a glyph. I modified the gene glyph for JBrowse such that if it looks at basically the transcript type when it's drawing the individual transcripts, and if it's a protein coding gene, it colors it one way, and if it's a non-coding gene, transcript. If it's a mRNA, it colors it one way, and if it's a non-coding RNA, it colors it another way, so that visually you can distinguish them in JBrowse.
Okay, thanks.
Can I ask a quick follow-on question?
Sure.
This is Andrew Farmer. Why not use multi-valued attributes for that situation?
I don't know. I hadn't thought about it.
Okay.
Basically, the part of it is the cultural inertia that I talked about before. Is that this is the way Wormbase had always done it before I started working at Wormbase, and none of the other NIH mods had dealt with this sort of problem in an explicit way. So basically, I said, well, why don't we just do it like this? And everybody said, okay.
Okay, fair enough.
Yeah. And then finally, the last thing that we specified is that, except as noted anywhere else in the specification, that there are no restrictions on the tag value pairs in the ninth column. They'll be ignored, but they can be there. So if you generate tags for somebody else to consume, which is a very common thing in Wormbase for sure, SGD does it a lot too, those can still be there. So they can basically generate the same GFF that they're making for the Alliance plus putting out into the world. So that was, I think, helpful to a couple of groups.
Is that still? Oh, sorry. Can I ask another question?
Go ahead.
We're getting the GFF3 nerds down here. So for the last one, I mean, so in GFF3 parlance, there are reserved attributes and non-reserved ones, right? So your tag would be capitalized if it's reserved and not if it's not. So is this restriction or no restriction guideline also count for the reserved attributes, the reserved tags?
Oh, yeah. Well, I mean, so for the reserved tags like ID, parent, I forget off the top of my head, what other ones would be kind of structural, indicating this thing belongs to that thing. Then there would be, those would have to be used the way they're supposed to be used. But for other tags like ontology term, DB, XREF, things like that, they can be present, but they'll still be ignored.
Okay.
So yeah. Okay, so just to kind of talk about a few of the positive things associated with doing this. Basically, it has made building a consistent JBrowse instance for all the species easy. I'll put in quotes because there's, in life there's always things that get in the way. But it has gotten remarkably easy. I can actually script most of it, and it usually works. And this ease has been made easier still by very recently we Dockerized our JBrowse server. And so now going from release to release, I think is going to be really easy in that regard as well.
Scott, this is Nathan.
Yes.
Sorry, just a quick question. Do you have a link to your Docker? Is there a link to this JBrowse instance on Docker Hub, or do you have a Docker file somewhere in the Git repo you can add to the docs?
Yes, I will do that. I'll add. So I sent the link for this presentation to Jacqueline, and so it's probably getting posted somewhere. I don't know, but I will add to this slide. I'll add a few links both to GitHub and Docker Hub. I should have done that, but thank you for thanks for suggesting that. We also generated a written standard, which I did think about putting the link into the presentation, so that's good. And there's actually, you can tell it is still a living document. There are tons of comments in it. It's a Google Doc, and there's tons of comments from people. This spec grew over quite a while, probably a year of conversations about it. And so for the moment, that's mostly it in terms of positive results. I mean, basically, for the Alliance, GFF is not an input format. It is just something that the individual mods create so that we can create JBrowse. The input format into the Alliance from the mods is a JSON that they created a spec for separately. So this standardization is kind of a small piece of what the Alliance is doing compared to all the other data that's being dumped out and put into the Alliance data store. But anyway, regardless, it's still a good thing. On the downsides, there's also not much in terms of major downsides. There were some minor growing pains, initial confusion between groups, as we talked about, standards development sort of stuff. There is one thing that disappoints me and some of the data quartermasters at the Alliance is that there's no validator yet. Everybody agrees that we should have one, but nobody has the time to write it. So basically, the validator is me when I try to create JBrowse and I say, oh, you know what, that didn't work, which is not a great validator because JBrowse is pretty permissive as to what it will allow in. So that's not great. Also, the other downside is that every mod did have to spend some time rewriting their GFF creation scripts. And for some groups, it was worse than others, just because that's the way life is. And when two groups do things in a very different sort of way, there's going to be some meeting in the middle, but usually it's going to be closer to somebody than the other. So that's just life, I guess.
A quick question, Scott.
Yeah.
Sorry about that. As far as the validator goes, are you talking about just validating the correctness of GFF3 or the conformance of a GFF3 file to the specification?
Yeah, really the conformance of the GFF file to the AGR specification, which shouldn't be terribly difficult. Go ahead.
Yeah. Have you used Genome Tools, GFF3 validator?
I have used that, yes.
Okay.
And I've thought about extending it. Yeah.
I see. And I think there was another one, Andrew, that you sent an email out about the other day. I forget.
Yeah, that was Monica's group's GFF3 toolkit, which looks really promising.
Okay. It's probably useful to include links to both of those in the docs.
Will do. Happy to advertise it.
Great.
But yeah, as Scott was saying, that there's one, it's hard to write these validators. I mean, our group did one, but we did it with our problems in mind, right? I mean, part of the problem with such a nicely flexible format is that you can break it in an infinite number of ways. So how you decide to evaluate those breakages can be challenging. And what it seems like what Scott is looking for here too is to specifically validate the AGR requirements.
Right.
So.
I see. So like not just a syntactic, but like a semantic type validator.
Yeah, exactly.
Yeah. Cool. Thanks.
So anyway, so that's basically it. I just wanted to thank. So Nathan Dunn, which I imagine many of you know, who works on Apollo, he has worked a lot with me, as well as Paul Hale, who's at MGI. And then those are the names of all of the data quartermasters at the various groups who helped put together the spec. Some of whom are no longer working as data quartermasters. They've moved on to more important things. So anyway, yeah, so that's it for me. If you have any other questions, we can talk about it, or we can move on to talk about other people's woes.
Cool. Thanks, Scott. Any other questions right now? I think if there aren't, maybe we can move on to a different example. I think this is has a couple more questions for me, but it'll just drive discussion in a bit. Okay, so maybe I'll go back to sharing these slides again, but here. Okay, so Chris, would you like to talk a little bit about some of the issues that you've seen?
Yeah, I mean, I didn't really prepare for this, except for I just knew I was going to say something. So,
No worries.
so I think like some of the things, I think most of our problems currently have to do with parsing stuff from NCBI because they change every so often. And some of the, I mean, just thinking some of the things that I might have come across in the last couple of years, but they might have changed by now, or I feel like they're not always consistent in what they're going to use as the SO term for a transcript if it's not just a regular protein coding gene, or like, you know, they'll use transcript and mRNA for things that are protein coding or long non-coding, but then when it comes to other kinds of RNA, they'll not be consistent on whether they're going to have a transcript parent or they're going to just call it something, you know, a more specific term. And so like when we process many species, even I feel like even there, depending, it just happens to do with the date of when they generated the GFF for that organism, I think. And so it's not even consistent when we're processing a large number of things. And so there's that one. Then they also, I feel like we have a lot of problems with the pseudogenes that we get from them. And sometimes I think like a lot of times we like just eliminate the pseudogenes from the GFF because they're so inconsistent and cause so much trouble. But I think the like representing pseudogenes is probably just another, like a future topic to be developed, because I don't think there's been enough kind of standardizing of how to represent those. And then another thing that I know that I prefer for my computational, like some of the work that I might do in research where I'm doing something to, like, I might want to understand relationships of different genome features with each other. And I use intersect bed a lot, and a lot of times I'll want to use intersect bed on exons for all the different transcripts. And it's just a lot easier for me if each RNA or transcript has its own set of exons, even though that exon might be shared in two different transcripts. For example, like I guess like the kind of official GFF3 that Monica had on her slide, there was an example of either exon or CDS. I guess it's probably exon that had one exon, you know, it said parent mRNA or parent whatever equals, and then it listed off 3 mRNAs. And so that's more difficult for me to deal with than if each individual mRNA just had its complete set of exons, even though some might be redundant. Then I thought JBrowse required that too, but maybe I'm wrong. And then, okay, so that's kind of, I think, things that I've found with GenBank or NCBI. And then just kind of it's been a while since we've actually taken in a new official gene set generated by a research community. But back when we were getting some different GFF, it's mostly I don't think Maker has this problem, but GFF that used to be produced using Glean didn't have a gene line, so there'd be an mRNA line or something. So everything would be given as at the transcript level. And so we would get GFF where there weren't genes at all, although it was clear that many of the RNA came from the same gene locus, but the IDs, or there's nothing about the GFF that would actually indicate that there are multiple RNAs from the same gene locus. And so we would have to compute the gene locus, and then we would assign IDs that reflected the gene locus. And that was that. So in Hymenoptomine, we have gene alias or transcript aliases that use, map our new IDs that would consider genes versus the original ones. People weren't really thrilled about that, but I think usually it's easier for people to interpret their results if they understand, they get a whole list of transcripts and they can know which ones are actually just different transcripts of the same gene. So,
right.
Yeah, I think that covers it.
Thanks, Chris. Yeah, I've nodded my head vigorously to most all of those problems. Okay, maybe we should move on, Andrew. And I wanted to mention it's 35 past the hour, and it would be great if we could sort of wrap up the examples in 10 minutes or so. So yeah, not to cut everyone short, but I guess we might need to cut people short. So we'll see. So Andrew, go for it.
Okay, well, so 10 minutes for everybody to present their examples, is that?
Yeah, since we wrap at the top of the hour, and there might be some time needed for extra discussion afterwards, or we might need to do another session of this, we'll see.
Okay, well, I didn't really prepare much that was formal, but I did have this example. I think the thing that, I mean, everything that has been said so far, I have also nodded my head vigorously to. The one thing that I think is kind of interesting about GFF that plays into some of these issues is the multiplicity of ways that it has to identify things. So this, the mathematician Poincaré once called mathematics the art of calling different things by the same name. And I think you could characterize biology as the art of calling the same thing by different names. And so we know that there are these kind of standard attributes for ID and name, as well as alias. And in this particular example, which is actually an example of some problematic structural annotation that formally isn't wrong, but basically what happened in this GFF that I was given to try to process, and all I really wanted to do was to get the proteins out of it. And what they had done was to basically give a different mRNA feature as a parent for different exons and CDSs. And technically there wasn't anything wrong with it. You just ended up getting a lot of little peptides instead of what you really wanted, which was the combined peptide for everything. But one thing that we face a lot is that it's not exactly clear what the distinction should be between IDs and names. And oftentimes they're the same, like in this example. I think formally the spec sort of suggests that IDs need to be unique within the file. But they're maybe not even really supposed to be used outside the context of the file. They're just kind of internal keys. Although the uniqueness isn't really enforceable, because there's this weird thing where you can have multi-features that share the same ID. And then name, I guess, is sort of supposed to be the thing that labels things. But if you're translating a GFF file and trying to get FASTAs out of it to represent the features, which one of these is actually supposed to be used in the FASTA headers for those files? Is it the ID? Is it the name? Et cetera, et cetera. In this particular example, which I won't go through in any detail, it turned out to be the alias, one of the aliases that needed to be used to sort of figure out how they were actually intended to be grouping their exons into each isoform. And this was an example of kind of a new Arabidopsis genome that was getting annotated in the context of pangenomics. And you'd think it would be easy to annotate an Arabidopsis genome, but apparently new pipelines being developed don't respect any of the rules or break them in the same way that we're used to. So this particular example was just a case of trying to fix up somebody else's misrepresentation of structure and having to use these sort of clues being given by the various ways of identifying things to try to get their structures back into place. And I guess I'll just leave it at that to give other people time.
Great, thank you, Andrew. I was also nodding my head vigorously with the FASTA headers. Okay, Nathan, would you like to talk about anything?
I'll make it brief, but just to add to or comment on something that Andrew mentioned. The ID, if a feature doesn't have a child or is not part of a multi-line feature, then the ID is optional. And I generally exclude that if I can get away with it, just because when you're trying to store GFF in an efficient format so it can be rendered in a browser like gbrowse or jbrowse. It helps to minimize the file size, which is a reason why I typically exclude the source column as well, or just, substitute it with a dot. But I submitted a pull request to BioPerl a few years ago to not store the ID after a GFF is loaded in the BioDB feature store module. So I guess that's my opinion. IDs are not necessarily required, and I think if you export a GFF using, now the name of the BioPerl script escapes me, from a BioDB feature store database, then it just generates IDs, and they don't necessarily have any meaning, but they're correct for features that span multiple lines or for features where there's a hierarchy. The other thing I was going to mention was about the GFF3 validators. That was just kind of a tip. And for JBrowse, maybe Scott's experience is a little different, but it helps to, when rendering GFF in JBrowse, it helps to use TABIX indexed BGZipped GFF3 files to reduce the file size. And that seems to render relatively efficiently. That's all I had.
Cool, thanks, Nathan.
Yeah, I actually, for the TABIX indexing, I experimented with it a few years ago, and I think there was something that it didn't work the same way as like the NC list, and so I got away from it and basically never went back because
it doesn't.
I suspect that's been fixed since then, but that's fine.
How do you represent? So out of in Apollo, is that coming from a Chato database then, or I forget how that works?
No. You mean the way we use Apollo in AGR?
Or how do you represent? We don't use it. Yeah, JBrowse.
Yeah, we don't use it for. Go ahead.
Well, I was going to say. So then, how do you render features? How do you take your GFF files and render them in JBrowse?
We use the command line scripts that flat file to JSON, so it goes into empty lists. Yeah.
Yeah. So that can be quite inefficient because if you have a lot of tracks, I mean, that can just create lots and lots and lots of little small files.
It does. That's true.
Yeah, quite inefficient compared to a You should give the TABIX index BGs of GFF3 format another shot.
Yeah,
Nathan's trying to say something.
Okay, can you hear me? Sorry, I wasn't sure my microphone's in it. So one of the reasons that we don't use that as much, I really wanted to use the TABIX index for other things that we were doing with Apollo. We do use them for things like VCF, but the issue is it's hard to get histograms out of the TABIX files because the ranges are so large and there's so much data. It becomes really inefficient when you get lots of density and lots of chromosomes to have an efficient way of doing that. And really what we should do is put another layer of indexing on top of those TABIX to use those, because I think both Scott and I would agree that's what we prefer to use. But having, if you want to have a density histogram with the GFF3s, you have to use the NC list.
And is the flat file to JSON more efficient for that?
Can I cut this discussion short, because I think this isn't about the GFF3 format, but about the JBrowse.
Right.
Sorry. I don't mean to, it's great to discuss, but we need to move along. So Nathan, did you want to talk about anything else with the format specification?
No, that's all I got.
Okay, sorry, two Nathans here. Nathan Dunn, did you want to say anything too?
Overloaded.
Yeah.
No.
Okay, thank you. Maggie, did you have anything you wanted to contribute?
Yes. So this discussion makes me think that AgBioData has an excellent opportunity to sort of guide how GFF3 files are formatted in not just the ag community, but outside of the ag community. I'm thinking just as the FAIR data paper I think made that discussion very broad and helped for people to understand what FAIR is and how important it is to make their data fair. I think AgBioData might have an opportunity to perhaps write a paper on this, maybe decide on what a GFF3 format should be, write a paper on this in the hopes that we can have some of our thoughts disseminated, beyond this group, and help people to sort of understand what the issues are, and that there are ways of formatting this that are probably going to be useful for most people. Although this discussion here tells me that not everyone, even within this group, there are people who have different ways of doing GFF files for their tools. So to the extent in which we can streamline this, I don't know. But I'm also thinking too, it's probably useful, again, I don't know who's going to write it, but to make some kind of tool that doesn't just validate, but will actually modify existing GFF files to conform with a standard that this paper or this group develops. I think that's kind of my overview on sort of how to address some of these problems, and that's all I have to say.
Awesome, Maggie, thanks. So those are really good points. Okay, so we had some slides from Philip Bayer. I'm going to do those very quickly. And so maybe to put some interpretation in here too, it seems to me like there's two sets of problems we're having here. One is people want to use pre-existing GFF3 files that come from a given tool and or database. The second is we're also databases here, and we also need to generate GFF3. And what problems do we have in making decisions to generate the same things that our downstream users can use without too much effort on their own. So it's both kind of in and out. And Philip's comments seem to be along the using lines of using existing GFF3s. So yeah, Augustus seems like it's similar to Glean, Exonerate, yep. And yeah, we do find many broken GFF3 files out on the internet. And here's a prominent example of one. So yeah. Okay, and I had some from i5K Workspace, but I don't want to dwell too much on these because it's been similar. Maybe just to say we deal with a lot of manually generated GFFs through Apollo, but also through other means, and there's just an infinite number of ways to break them, which is why we wrote this GFF3 toolkit here. One thing that I will add that I don't think I've heard bring up so far is that NCBI could, so Chris pointed out earlier that when you retrieve files from GFF3 files from NCBI from different date points, they are formatted differently, and you have to accommodate your scripts to account for that timing when NCBI released them because they change. But they do also accept GFF3 files, and maybe this could be an opportunity to, or a standard to rally around, because they do have their own formatters. They do have ASN validate, so you can transform GFF3 to ASN with their tools. And validate them using ASN validate. So there's stuff out there, and maybe we don't need to reinvent the wheel if we can all just agree on one thing like AGR has done. Okay, so possible solutions. So I wrote a couple of things up here about, are there ways to standardize representation, kind of what Maggie was just talking about. But I wanted to start off the discussion with some of the things that occurred to me as people were talking here. So especially with when Scott mentioned, well, the mods just use GFF3 for JBrowse. And so that clearly defined for you guys what you have your 5 mods, you have your 5 ways of representing GFF3, and you have your 1 tool to slurp it into. That kind of cuts down on the number of parameters you have to deal with, right? It's just for JBrowse. So Scott, maybe you can comment on, do you know why the mods don't use GFF3 for ingest into the database? Is it a historical reason, or are there some fatal flaws with that format that mod databases have accommodated in different ways?
No, the reason we don't ingest GFF3 into the Alliance is because there are so many other data types that aren't covered by GFF3. So I mean, we're bringing in, you know, phenotypic data, we're bringing in disease data. So you can't represent that in GFF. So we had to come up with something else. And so the fairly reasonable solution that was considered there was, well, since we can't use GFF for everything, let's create something that will encompass everything. One thing we have talked about and may do in the future, I don't know, is because we're ingesting all this data in our specified JSON format, is it should be possible then to dump out GFF3 from the Alliance that is uniform. That is also that's a difficult task. So we haven't done that yet, but I think we may do that in the future. And then each mod won't be responsible for generating GFF for use inside the Alliance, which will be nice. I'm looking forward to that. But because then we'll have control over exactly what the GFF looks like, as opposed to having 5 mods generating it.
Right. So do people think, I mean, GFF3 seems to be one of the ways that the bioinformatics community has coalesced around gene annotation, but I mean, Chris Childers, if he's still on the call, he always talks about this one example when he was talking to an NCBI representative, and they were saying like, no, our ASN format is the standard. So are we misguided in making GFF3 the standard, or should we stick with it? What do people think?
This is Effie. I think trying to come up with a brand new way of doing this is likely to be a problem. People are so used to using GFF.
Yeah, and sorry, this is Chris. It's still like that story still kind of, amazes me because, you know, from his perspective as a data specialist at NCBI, it is the standard that they use, whereas like the rest of the world is like in a very different space. And it's tough because they still expect the rest of the world to realign to fit their systems. And I'm not saying whether one format is better than the other. I know there's like some really amazing things you can do with ASN but you know, that's not where we are as a global community.
Right.
Hi, this is Surya. I don't know if you can hear me.
Yeah, we can hear you. It's a bit broken up, so you might want to speak up a bit.
Okay, the only little thing I would want to add about ASN1 is that standard is probably tied in very closely to the NCBI C++ toolkit. And they have parsers and everything is built on top of that. So maybe that could be a reason why they don't use GFF. And as far as the rest of the world is concerned, because GFF3 is a simple tab delimited format, which doesn't require fancy parsers for the naive use cases. If we have extensions to GFF3, that would be more easily usable than something completely different. I think.
What do you mean by extensions to GFF3?
Oh, in terms of, like the key value conventions or the new data types, if you're trying to handle new data types within that layout. And the idea about having a single file format for every single type of data is a little complicated, but I don't really want to get into that discussion. That's not about GFF3.
Yeah. I was also amazed when Scott said that you could do that. Sorry, I was interrupting somebody. What was that?
NCBI does provide GFF3 files from like their eukaryotic genome annotation pipeline, for instance. So maybe they don't use it internally for everything, but they at least provide some of it to the public.
Right, exactly. And they do accept GFF3 files if you want to submit gene annotations to them. They have tools that transform the GFF3 to ASN format that they can then run their NCBI plus a toolkit that Surya was talking about. So
and I have to say the NCBI GFF has gotten a lot better. I've talked a lot of times with Terrence Murphy at NCBI, and I was pleasantly surprised yesterday when I was doing some coronavirus work, and the GFF for the coronavirus was actually really good. It was like perfect. So I was really happy about that.
Good. So maybe NCBI could, since AgBio data is a broad set of organisms, we might still be able to rally around NCBI upon evaluation. Okay. So right now we kind of need to figure out where to go from here. We have only about 5 minutes left, and it seems like we have some good input, but there's still a lot of questions to be asked. So what the exact problems are, what the tools are that we need to ingest for. So in developing an ag biodata standard, how do we find consensus across all of these different use cases? So maybe what we need to do is, if we had a working group, oh gosh, we have so many ag biodata working groups, but we could try to consider something like an ag biodata working group where we try to hammer out, okay, we need to find a convention for either modifying tools from other exports, or for ingest in our databases, or for a presentation from the databases to our downstream users, and figure out the most pressing needs and identify a standard around them. And maybe start with existing standards such as what Scott described for AGR's JBrowse ingest and NCBI standards as well. Maybe we can rally around that.
This is a very timely thing, because I'm finding GFF files in many, many different formats right now. And just listening to people talk, I learned about a bunch of tools I didn't know exist. So it seems like finding a way to corral what's out there would be a good thing, a good place to start.
Yeah. So we'll probably have to see what to do, and maybe pursue Maggie's idea of a paper. Since our group has already written a toolkit to do some validation, maybe that would be something that we could take on. We'll have to see if we have the incentive and development expertise for that. But so I think we should stay tuned. And so I can say that I will, at least for now, take the lead on figuring out if we can do something with an ag biodata based on the information that we collected here. And so I will probably ping some of you guys directly to see if you want to work on this, and maybe also send out an email to the ag biodata listserv to see if there's enough of an interest group to work on something, and maybe have 1 or 2 or 3 short but effective meetings where we try to hammer out a standard, or just say, we're going to reuse what AGR did. It's cool. We don't need to do any more work. We're done. Perhaps that's it. So does anybody else want to comment on that?
I think that
one thing that go ahead. Okay, I was just going to say one thing that I wanted to.
Scott, go ahead.
Okay.
You were louder.
I'll go. Yeah. The one thing I just wanted to add is that I wanted to comment on what Chris said a while ago. Chris Elsik said about pseudogenes. We have very specifically put off dealing with pseudogenes because they're gigantic pain in the ass. Yeah, so I have no idea how we're going to deal with them, because I don't like any representation of pseudogenes in GFF, but I'm going to have to come up with something we like.
Yeah. I actually, so if to add to that, if you look at the INSDC's pseudogene qualifiers, that's actually the best thing I've found so far that seems to relate to the biology of the origin of the pseudogene as much as possible. So it says like kind of where it's coming from. And I think there was like somebody was trying to develop a pseudogene ontology. So there's some stuff out there, but there's no standard again, except for NCBI has their own internal standard. Cool. Sorry. Can I post the link for the link? Yeah, I'll post a link for the pseudogene standards. Sure. Sorry, somebody else was talking while Scott was talking. Go ahead.
This is Pankaj here. And I don't know if it came out in the discussion or not, because I got involved in another sidetracked on that thing. Obviously, GFFs are mainly for the gene features that are mapped on the genome coordinates on that. But a lot of Plant-D folks, for example, have mutants and QTLs and phenotypes also being mapped on these tracks for doing a lot of application stuff. And so far, I think we have hacked the GFF format for putting that data in. But if I don't know, Scott and others, what are your feelings about from the Genomics Alliance, or how do we formalize more about standards about bringing in phenotypes mapped on these maps in the GFF format?
Oh yeah, I don't have an answer to that. Yeah, I don't know.
This is Chris Elsick. So we, I guess I thought a little bit about that issue because we have QTL in our Intermind databases, and then we load them as GFFs, and so we make sure that we're using a sequence ontology term for column 3, but I can't remember what it is. But that's, I mean, at least like for the QTL, we at least our data was pretty simple because it was, you know, it didn't have children. So, but you know, there could probably be better ways of doing it. But I would say one thing is to just keep, you know, pay attention to sequence ontology.
Yeah, and actually, I guess the other thing I should add is that we probably addressed this somewhat at Wormbase, not at the Alliance, but we probably do it at Wormbase. But off the top of my head, I can't think of anything that is there. So if you wanted to look at what we've got at Wormbase's JBrowse, and if you have any questions about how I do that, I would certainly be willing to answer them. But I can't give you anything useful right now.
All right. Yeah, we'll take a look at it. Thanks.
Yeah.
All right, we're past the top of the hour, so we should probably wrap it up. But thanks to everyone for participating. Hopefully, if anything, this has given at least those who work with GFF3s a list of links of other stuff that you haven't heard about before, and that might be a good outcome regardless of what else happens here. And let's keep the discussion going. Thanks all. Talk to you later.
Thanks. Bye.
Thank you.
Bye.
Bye-bye.
Thank you.
Thanks. Bye.